And so the next speaker is Vera Gorbunova from University of Rochester. She's going to talk about epigenetic rejuvenation with SIRT6. Right, thanks great to have you here, Vera. Thank you.
Okay, thank you, Peter, and thank you all the organizers for inviting me. And I've been really enjoying. Oh, sorry, I have to start again sharing. I was really enjoying the talk. So this topic is really so interesting to me. Okay, advanced.
Can see your screen okay, but it's in
yes, I'm just trying to get to the presentation mode.
That looks better.
All right, almost there. Okay, so there were great talks the first day and yesterday, talking about aging as being the product of so-called epigenetic or epigenomic drift. So that is how I imagine epigenomic drift to look like. So something very sad that is happening, drifting away, and perhaps that results from a combination of factors. So if this is the young cell with the chromatin being nicely organized into compact heterochromatic domains, more open domains, epigenetic landscape is very sharp and defined, but over time, when the cell goes through DNA replication, transcription, and of course DNA repair, things really get messed up. Even the cell is trying very hard to put everything together the way it was, but it's not perfect. So over time, the noise accumulates and the epigenetic landscape becomes smoothened. This is also my favorite analogy to the socks in a drawer. Once they are folded neatly in the beginning of the week, it looks very nicely. So that's our chromatin in the young state. But after you go in and out and pull things, it starts to look like that, even if we are trying to put it back neatly, but it still doesn't work. So, of course, the big question is how to reverse it, how to go from this drawer to this drawer. And that will be the subject of my today's talk. There are several ways of doing it. And of course, nature figured it out because it happens every time a new life is conceived. So when the baby is born, its epigenome is new and the age of 0. It's not the average age of both parents. So obviously there is an evolutionary process that can take care of it, but that gives rise to the next generation. And what we would like to achieve is to apply the same strategy to the somatic body. All right, so from this introduction, so this is the gene we've been studying for a long time. It's called sirtuin-6. Raul Mostaslavsky gave a talk on the first day focusing on sirtuin-6 and primarily its role in cancer and metabolic functions. So SIRT6 was called longevity sirtuin because of all sirtuins, sirtuin-6 has the strongest phenotype that's related to aging. If you knock it out, you get these mice that are very defective. They have some progeroid features. They have a lot of genomic instability, and they die by 30 days of age. So this is the work from Mostaslavsky when he was a postdoc. degenerated these mice. And then Chaim Cohen's lab generated mice overexpressing SIRT6. And these mice live up to 20% longer, which is quite amazing. So our group has contributed a lot in understanding the role of SIRT2 and 6 in DNA repair. So here is just a big summary from a recent review. So what we and others showed that SIRT6 is recruited to the sites of double-strand breaks. And it sort of prepares a platform for other factors to come in. And this is why it is upstream of multiple DNA repair pathways. So it plays a role of like probably cleaning, sort of preparing the platform, changing chromatin at the site. It also recruits PARP1, activates PARP1, and then recruits other factors that are important for DNA repair. So this is one function of SIRT6. My lab is also well known for the studies of unusual model organisms, although in today's talk I will not be talking very much about it, but we study various long-lived creatures from naked mole-rat to bats and even bowhead whale. I could give you a whole different talk about bowhead whale. And the goal is to understand the longevity mechanisms that evolved in these species and then try to see how to apply them to humans. So here we've done a little bit of that. We took a collection of various rodents with lifespans ranging from 3 years to 40 years. We measured the efficiency of double-strand break repair, and we found it to correlate very strongly with maximum lifespan. And SIRT2 and 6 was the major determinant of this correlation. So it's been published in 2019, so I'm not going into details, but I just want to emphasize that what we found was that SIRT2 and 6 activity correlated very strongly with maximum lifespan of the species. So that was very interesting. And then we also discovered another role of SIRT2 and 6 in epigenome maintenance. So this one is DNA repair, but it is also important in epigenome maintenance, which sort of parallels the role of yeast SIRT2, where it is responsible for gene silencing and for reinforcing heterochromatin. So what SIRT2 and 6 does, it also maintains heterochromatin, and it is important for silencing transposable elements. In these poor mice that are missing SIRT2 and 6, what happens, the heterochromatin really unravels. Their centromeres open up and telomeres open up, and transposons become very activated. So that is maybe another reason for their severe premature aging phenotype. So if this is what the chromatin looks like, a very large part of our genomic DNA is occupied by transposons, these endogenous parasites that, well, there are lines and signs that make about 30% of the genome, but then also DNA elements, endogenous retroviruses. So everything altogether is more than 50% of our DNA. So maybe our chromatin looks like that with all of these parasites sitting there. And of course, thinking just about the volume of these sequences, they must be playing a role in epigenetic reorganization with aging. So this is a picture from an old review that we wrote where we proposed that with aging, line 1s get activated, so they're like sleeping dogs. And when we are young, everything is fine because the cell has multiple mechanisms to suppress these elements. But with aging, they awaken. And although initially we believe they just cause genomic instability by the virtue of jumping around and cutting DNA, but what later turned out, they also trigger inflammation, which is one of the hallmarks, or new hallmarks of aging. So what specifically SIRT6 does in this, and this is published several years ago, it binds to 5' UTR regions of line 1 elements, and it recruits CAP1 and HP1, packaging them into heterochromatin. So this is what happens in the young tissue, but in old tissue, SIRT6 no longer binds there very well, so that contributes to activation of these elements. What we demonstrated was that with aging, line 1 starts to accumulate in the cytoplasm in a sort of cDNA form, this cDNA creates a problem because it then recognized by SIGA-STING pathway and drives inflammation. So we were able to reverse this by giving mice antiretroviral drugs, and we could make those knockout mice live longer. So there were these 2 papers published back-to-back, ours in Cell Metabolism and John Sedgwick, our collaborator in Nature, showing this mechanism of inducing age-related inflammation through activation of reactivation of line 1 transposons. So that's sort of the pathway that cDNA is formed, triggering SIGA-STING signaling, and that can drive multiple age-related pathologies because inflammation may be the root cause of diabetes and Alzheimer's and cancer. So pretty much all age-related diseases have inflammatory component in them. So these are the 2 functions of SIRT6. And at that point, we became interested, since we know that there is a relationship between SIRT6 activity and maximum lifespan, is there a relationship within species, for example, among our own species, humans, if long-lived individuals have more active SIRT6. So this is a collaboration with the group of Yusin Su at Columbia University, and Yusin studies human centenarians, and Matt Simon was a postdoctoral fellow in my lab. Now he moved on to his independent position at Jackson Lab. So we got together with Yusin, and she was sequencing human Ashkenazi Jewish centenarians, and she discovered several alleles in SIRT6. One of these alleles was enriched in centenarians, so that was quite amazing. So this allele had 2 missense mutations in the C-terminus of SIRT6. Because that was a missense mutation, so we could engineer it and test functional properties of this protein. So first we looked at its enzymatic activities, because SIRT6 can deacetylate its substrates, which also includes some histone substrates. It can also mono-ADP ribosylate substrates, and this is the activity that was neglected for a long time, but we discovered multiple targets, including PARP1 and CAP1 and histones. So this activity is very important for epigenetic component of SIRT6 function. And then we tested the centenarian SIRT2 and 6. To our surprise, so here we are measuring the acetylation activity. The centenarian variant, and here we have 1 mutation, the second and cent is when both are together. Centenarian variant had actually weaker deacetylase activity. We didn't expect it. We thought it should be better in like every respect, but no, it still had deacetylase activity, but it just was not as strong. But its other activity, mono-ADP ribosylation, was enhanced dramatically compared to the wild-type human version. And then functionally, when we tested it in DNA repair assays, centenarian SIRT6 was better. So this is the wild-type stimulation, and this is with centenarian. So it's really better at DNA repair. It was also better at line 1 silencing. So here we are measuring line 1 expression. When we overexpress in the wild type allele, individual mutations of both together, and you see that it can more efficiently silence line 1. So it seems to be a better enzyme, at least with respect to these activities that are important for epigenome maintenance. We also looked into the interacting partners of SIRT6, and we were hoping to find some differential interactors between the centenarian allele and the wild-type allele. They all interacted with the same protein, so we didn't find anything that would interact with centenarian but not with the wild-type. But there were some proteins that were interacting more strongly with centenarian version, and that was lamin A was the best hit here, which is kind of exciting because lamin A provides the scaffold for chromatin in the nucleus. It was known previously that SIRT6 interacts with lamin A, and now we see that centenarian version interacts much better. So lamin A is a very famous protein. It's associated with multiple human genetic syndromes. Perhaps the best known is Hutchinson-Gilford progeria, in which case lamin A is defective, so it results in premature aging. But there are also some polymorphisms with lamin A that are reported in centenarians. So I guess it could be, have worse function, or it could be improved to provide better structure. So what we hypothesize is that perhaps the centenarian version of lamin A, by virtue of binding more strongly, of SIRT6, by virtue of binding more strongly to lamin A, is somehow better at maintaining heterochromatin or chromatin organization altogether. And it is also interesting that it has a different ratio between the two enzymatic activities that is more beneficial to longevity. The acetylation activity is essential, so you cannot go successfully through development without this activity. So it's not to say that you don't need this activity, but maybe what we speculated in adult organisms just for the maintenance function, while this is important for development, but for maintenance, this activity may become more important for an adult. So, okay, this is all very good. In centenarians, they are lucky if they have this allele. It's a very rare allele, so not all centenarians have it, of course. But what about, most of the population who is not lucky enough to have this allele? So we started thinking, is there any way for everybody to benefit from more active SIRT6? And this is the work of Min-Soon Lee, a graduate student in my lab, and Zhizhong Zheng, who is a bioinformatician postdoc. And this is in collaboration with Steve Horvath and Ken Raj. So I'm going to show you unpublished data now, which we are wrapping up, and we are very excited about this. So what Min-Soon did, she took primary human fibroblasts that Ken Raj isolated from people of older age, 60 plus up to 90 years old, and they were very low passage, so they haven't yet forgotten where they came from. So we took those cells, and then Min-Soon transfected these cells with lentiviral vector expressing SIRT6. And she did it for a fairly short time, just 2 weeks. And then we applied Horvath's clock to see whether it changed methylation age of the cells. And to our amazement, in 9 out of 10 donors, methylation age went down just after 2 weeks of SIRT6 overexpression. So then based on we've done RNA-seq on these cells, and then based on Zhizhong's analysis, we see that the functions that were changed in these cells after SIRT6 overexpression are mostly related to anything that has to do with chromatin, like DNA conformation change, chromatin assembly, DNA packaging. So all of these functions we would suspect SIRT6 to participate with. We also analyzed biotoxic, and we found that some of the functions were enhanced and kind of became more open, some of the genes. So the Go term that was significantly enhanced was SWI/SNF superfamily. So which tells us that it's not that SIRT6 enzymatically does everything, but it perhaps affects other epigenetic enzymes and somehow changes heterochromatin together with others. So maybe SIRT6 has a more regulatory rather than directly enzymatic role in instituting whatever change it does to reduce epigenetic age. We looked at histone modifications. We see increase in H3K9 trimethylation levels. upon SIRT6 overexpression. And then we decided, okay, let's look at transposons. What happens to them? We know that SIRT6 is important for silencing transposons. So from RNA sequencing result, what we see that transposons are significantly silenced, and all of them, lines and signs and even DNA transposons and also LTR transposons. So it reduces expression. Then from attack sequence, we see that transposons show reduced accessibility, again, all of these classes. Well, I guess with exception of sign, but the other three classes show reduced accessibility. And then we also analyzed DNA methylation on transposons using MEDIP, so precipitation of methylated DNA and sequencing. And here we see that all classes of transposons massively gain methylation upon SIRT6 overexpression. So it really takes those transposons and packages them back into heterochromatin where they belong. So that is all exciting, but you know, this is the type of intervention we tried on these cells is a gene therapy, which of course is possible, but it would be preferred to use some kind of pharmacological approach. And there was a you know, effort going on to identify SIRT6 activators, you know, just based on the findings in mice and lifespan extension. So there were several molecules identified that activate SIRT6 deacetylation activity, because that activity was the easiest to screen for. So we just took those published molecules that modulate SIRT6 activity and tested whether they also enhanced SIRT6 ribosylation, because now from the centenarian study, we know that this is the one we want to enhance. So strangely, most of these molecules do absolutely nothing to mono-ADP ribosylation, which means the two activities are separate. You cannot stimulate both with the same molecule, with the exception of one molecule called Fucoidan that was first identified by Ruin Moadel from NIA. And that was reported to be a very strong activator of SIRT6 deacetylation, and we see it to be also amazing activator of SIRT6 mono-ADP ribosylation. So I thought, okay, let's look into this molecule. So this molecule isn't something that perhaps you know, medicinal chemists would be excited about, because it's a natural product and it is a polysaccharide. So it is derived from brown seaweed. So you can see these various versions of it. So it's not a very defined structure. So it's a mixture of monomers of fucose that can be connected together in various ways. It can be linear or branched. It can be also sulfated. So some preparations of Fucoidan would be more active than others. And we decided, okay, let's try to see if Fucoidan, because in vitro it's such a strong activator of SIRT6, is it going to do anything in vivo in mice? And this is the work of Ali Biashada, a technician and excellent lab manager. So what Ali did, he took some Fucoidan, and well, I want to also say that brown seaweed is a very popular food source, especially in Asian countries such as Japan, South Korea, that have the longest life expectancies. So of course we could only speculate that this is because people eat seaweed, but you know, because it is so safe, I can with clean consciousness say, okay, everybody, let's eat seaweed. It's good for you. Even if, you know, through SIRT6 or not through SIRT6, it's good for you. So what Ali did, he took a preparation of brown seaweed that we showed to be very active in vitro on SIRT6. And then he gave it to mice. So we started from sort of middle to older age mice at 14 months old. We gave them Fucoidan as a powder mixed with chow and also in water, and just kept these mice. We started noticing after a few months that there were some differences in the coat. So here are control mice. The Fucoidan mice seem to have much better coat. But when we apply the more comprehensive frailty score to these mice, and this frailty score is very similar to geriatric frailty score with grip strength and walking speed, we see significant difference. So mice receiving Fucoidan are less frail. And over time, of course, frailty score increases as mice are getting older, but the difference is maintained. So this is very encouraging. And right now we are setting up to do a human clinical trial where we want to give Fucoidan to cancer patients after they've been treated with chemo and declared cancer-free. So at this point, people really suffer from frailty that's induced by chemotherapy. So we'll be giving people Fucoidan and see if it relieves frailty, because most likely DNA damage induced by chemotherapy really wreaks havoc in the epigenome, and that would be nice to have some help from SIR2 and 6. So we'll see where that goes. So just to summarize, what I would like to say is that, yes, we have a lot of transposons in our genome. They must be exerting some cost on us, the hosts. And it's not entirely clear what is going on. Well, it is pretty clear that loss of heterochromatin is one of epigenetic changes that happen very consistently with age. And transposons become reactivated. But if you think about development in different stages, that is actually quite peculiar, because in ES cells or iPS cells, transposons are very active. But there are also various mechanisms that trying to minimize their negative impact. There are all sorts of pvRNAs and other strategies that degrade them. So then when cells differentiate, in young but differentiated cells, transposons are packaged very tightly and they're relatively inactive. And that's when there is a lot of SIRT6 bound to chromatin. With aging, situation starts to change. So in old cell or in senescent cell, it starts to resemble in some way the ES cell again, because chromatin changes, some things unravel, transposons become reactivated again. But that's a bad situation. So while this situation is very nice and tolerable in ES cells, it's not tolerable in the old cells. So we're still trying to understand how to put all of these factors together. And Vadim Gladyshev was talking yesterday about this rejuvenation that happens during gastrulation, but it also coincides to when transposons finally start to get silenced after the initial few divisions where transposons are very active. That at this stage, methylation starts to be reinforced on them. So there are all these processes happening in parallel. And of course, the question is, how do we go from these cells either back here? Well, this can be achieved by complete Yamanaka factor reprogramming, or preferably here if we want to rejuvenate somatic cells. So this can perhaps be done with SIRT6. So back to this picture that we started with. How do we rejuvenate the epigenome? So it can be done by Yamanaka reprogramming, but that would be more like taking this drawer, throwing all the socks away, and then refolding them and putting them back, which sometimes may have its dangers. Or we could be more conservative and just package heterochromatin, and this can be done with the help of SIRT6 or perhaps other epigenetic factors that would not change cell identity, but reinstate heterochromatin the way it was. So let me summarize. Epigenetic drift leads to loss of heterochromatin during aging. Transposons are activated. Transposons can drive inflammation through formation of cDNA and SIGA-STING pathway, because the cell thinks that these are infective agents, well, even though they sit within us. This can be rescued by various ways, for example, antiretroviral drugs, but this is already acting somewhat downstream after transposons got on the loose, you can try to sort of silence the signaling pathways or silence the propagation. And maybe the even better way is to package them back into heterochromatin so that they prevent their expression altogether. And this may lead to some degree of rejuvenation. So I would like to thank members of my group. I was naming people as I was going along. So the work I showed you today, well, the work of many people, Shao Tian, he worked on comparative DNA repair, Matt Simon worked on centenarian SIRT6, and the story of transposons. Ali did the work with Fucoidan and mice. Min Soon and Zhizhong work on rejuvenation, and all of this was done in collaboration with Andrei Sulanov, who is my lifetime collaborator. So here, all the group is at the Cold Spring Harbor meeting, and we do have postdoctoral and graduate student positions available. So if you're interested in working on this subject or on whales or bats, please get in touch with me. And we have amazing outside collaborators. Well, some of them were giving talks in this meeting. Vadim Gladyshev gave a talk yesterday. We collaborate with him and with Steve Horvath and John Sedivi's group. We collaborate on the subject of transposons and many others. And I'm happy to take questions.
Excellent job, Vera. Really great talk. So there's lots of questions coming in. We probably won't get through all of them. And I'd like to start with the first question. And that is, so SIRT6 is obviously an NAD-dependent enzyme, and there's a lot of interest in NAD supplements. So has anyone looked specifically at NAD supplements, NR or NMN, on specific SIRT6 activities, either ribosylation or deacetylation? I guess deacetylation would be the first one to look at. Have people looked specifically at SIRT6?
Well, yeah, that's a good question, Peter. Among the 7 sirtuins, sirtuin 6 is the least sensitive of NAD. It has a pretty good binding to it, so even a little bit of NAD is enough for it perhaps to keep it active. So it's much less of a subject of this fine regulation using NAD. So that's why I think that NAD supplementation alone may not be sufficient to activate SIRT6. So we have to really look for more specific activators.
I see. Okay, great. Thanks. So yeah, a bunch of questions. Marta Kovacheva has a very interesting question, and is wondering, I guess in a nutshell, whether the compromised SIRT6 deacetylase activity, is that likely to be functionally relevant? Is it perhaps even beneficial, or is it just some kind of irrelevant passenger effect to the important thing that's going on with ribosylation? And I guess as a follow-up to that, is then interested in the effects of fucoidan on deacetylase. Does fucoidan affect deacetylase activity?
Yes, it stimulates both. So it's a really very strong activator of both activities. Now to the first part of the question, whether is it functionally relevant or not. So in vitro, we see this reduced activity. In collaboration with Ben Garcia, we looked with a quantitative mass spec, whether there is a difference in vivo in SIRT6 targets, in the cells that have centenarian version of SIRT6. And in vivo, we actually didn't see it. So I think in vitro, when we measure kinetics, it's apparent, but in vivo, when SIRT6 has enough time to deacetylate whatever needs to be deacetylated, it probably still keeps up with its job. So it's interesting, we have other mutants with separation of function, and we have one mutant that has almost no deacetylation activity, and that results in much enhanced ribosylation and also enhanced DNA repair function. So maybe, having some reduction in this activity actually helps promote the DNA repair function.
Okay. So Zhizhen Do has a question. Do you want to unmute and ask your question, Zhizhen?
Oh, hi, Vera. Hi, Vera Do here. So I have a question about this concept about aging is associated with broad loss and disruption of heterochromatin. I know I had this argument with Peter Adams a while ago because we see in senescent cells, they have this very strong heterochromatin foci, the SAF, and so indicating that the chromatin, at least in subtype of the senescent cells, are actually more compact, and then they may actually have more heterochromatin. Right, so I wonder what's your comment on whether there's more compact chromatin or is a global disruption of the heterochromatin during aging or senescence?
Yes, that's right. I guess, maybe that was somewhat a simplification on my side to say, well, there's just opening. Yes, in senescence, there is a disruption. So there are certain things that, turn into more compact heterochromatin when they shouldn't. So, of course, various things happen. But with respect to transposable elements, that's mostly loss of heterochromatin. And I think that what you discovered together with Peter, these cytoplasmic DNA fragments that are formed through autophagy mechanisms, that's, I guess, yet another pathway that can trigger inflammation through DNA damage. So, of course, transposon is not the only pathway, but there are several, and there is another one through mitochondrial DNA. So we don't yet know the relative contribution of all of these pathways, but perhaps all three of them together, and maybe also the D loops, the transcriptional loops as well, again, through C-gasting. So there are maybe 4 of such pathways that promote age-related sterile inflammation.
So a related quick question to the previous question about the 36 mutation in the centurarian. Are there any trade-offs? For example, is there any defect in this centurarian as you see, like infection or fertility, or any trade-off as you are aware of?
Well, not that we are aware of. We know that they made it past 100, so they must be they must be in good shape.
What about their children?
Yeah, no, nothing apparent. I don't really know the health history on these people because, well, we just took these samples from the cohort, and we didn't have access to all of this information. We just know that they lived past 100, and they had a familial history of longevity. But based on the studies in SIRT6 overexpressing mice, they don't seem to have any problem.
Okay.
Thank you.
A couple of quick questions from the chat, from the Q&A. Marie Van Drom asks, does Fucoidan activate other sirtuins aside from SIRT6? Oh,
we didn't test it. I'm trying to remember from the original paper of Ruin, where I think they might, although, I'm afraid to say something wrong. I don't know. My feeling is they don't, because that was published as a specific activator, and I think Ruin probably looked at others as well.
Okay. And question from Sarah Elgin. Are there any trials underway using Fucoidan as an intervention for long COVID, which is obviously inflammation related? That would be perhaps interesting to try. Do you think?
Well, thank you for this suggestion. Yeah, we should definitely try to propose it, especially there is a huge unmet need, and it is a very safe treatment. So there are several papers of clinical trials conducted with Fucoidan in Japan, where for a long time it was considered to be a healthy supplement. It was shown to do a variety of things, which we could all potentially link to SIRT6, although we don't know for sure. For example, it was shown to improve response to influenza vaccination in elderly, pretty much like that famous rapamycin trial. So Fucoidan did the exact same thing. It also improved quality of life in cancer patients, which was, one of the reasons we decided to go through similar pathway with cancer patients. So it was just administered to cancer patients, and overall the survival and frailty scores were improved. So that was another small trial in Japan. So it has all of these health benefits.
Okay, great, thanks. Ognian has a question, has his hand raised. Ognian, do you want to unmute and ask?
Yes, thank you. And thank you, Vera, for the fascinating talk. CERT6 looks very fascinating. It's been around for a few years already. It's already, I think, relatively famous. It looks very promising, and especially with your new evidence, it silences transposons, it reinstates chromatin, and it has all these beneficial effects. However, I was thinking, do you know or have you looked at what might be the possible drawback or adverse consequence of overexpressing CERT6? Because I'm thinking if it was so universally beneficial, it is a relatively straightforward mutation to increase its activity or its expression. In fact, you've identified in centenarians a mutation that increases activity. So evolution, I would expect, would have selected that if it were universally helpful, and we should all be centenarians by now, but we're not. So I'm thinking there must be some hidden cost that we're not looking at, perhaps.
Well, I think your question is, it has evolutionary component, because the question is like, well, it's possible to, evolve a mammalian organism that would live to 200 years like bowhead whale, but we don't all live 200 years because lifespan evolution is very much dependent on sort of, genetic benefits. So like mice do not live 80 years because they cannot survive that long. There are too many predators, so they maximize their reproductive output in their first year of life. So to put it in other words, I think we don't all have super active CERT6 because there was really no selective pressure. We have just enough CERT6 to take us through our reproductive years and a little bit beyond. But if we want to improve it, then we have to do we have to modify the system, engineer it better, because right now it's the same like with DNA repair or epigenome maintenance. Well, we have just enough for what our evolutionary needs used to be in our evolutionary past. So I don't think, everything that hasn't been optimized is because there are some drawbacks, although of course you're correct, there might be some. So far I can't really name any, but of course we have to look carefully.
But sorry, as a follow-up to this, I don't know if anyone has looked, does CERT6 increase also reproductive lifespan? Because if that is the case, then, because if it is keeping organisms healthier for longer, it might also increase reproductive and lifespan, and therefore it should be also helpful for evolutionary success.
I don't think it was in the original Chaim-Cohen's paper. We are going to look into that. We now generated our own CERT6 overexpressing mice. And the reason perhaps it wasn't in the original study was that was a randomly integrated transgene. It wasn't even overexpressed everywhere, like where you need it to be, in germline. So right now we have mice with CERT6 in the ROSA26 locus, so we can express it in any tissue we want. So we are definitely going to address this question and see whether there will be benefit. Of course, one might imagine maybe not. Maybe if a lot of effort is going into maintaining perfect genome, maybe then not enough resources would be left for reproduction. That's also a possible answer to your question.
Yeah, I see. Thank you.